Alright, section 12.3 is going to start at objective 5 and go all the way through chapter or objective 12. So, here we're going to start talking about the second line of defense, and specifically within the second line of defense you have phagocytosis, you have inflammation, you have fever, and then antimicrobial peptides or proteins. And so phagocytosis is just really a cornerstone of the beginning of inflammation. And it is able to link both the inflammatory process and specific immunity. So, in order to have phagocytosis, you have to have phagocytes. Phagocytes are going to survey the tissue compartments. Alright, they're going to look for and discover foreign microbes, particulate matter, and then even injured or dead cells, and help in the process of eliminating these materials by ingesting them. And then, in addition to just physically eating them up, basically, it's going to help in that specific immunity, so extract immunogenic information. The professional phagocytes are neutrophils, monocytes, and macrophages. Neutrophils are the first responders against bacteria. If you have pus, alright, let's just skip down a very, or to the, almost the last point here. Alright, if you have pus, you have neutrophils. And generally that's going to mean you have bacteria present. So neutrophils in general, their purpose in life is to phagocytize. Phagocytize bacteria, kill them, and in the process they generally die themselves, and that is what pus is. So neutrophils, because they are everywhere really, they're going to react very early in the inflammatory response. And a high neutrophil count in the blood, if you do a blood count following, or if someone has a fever and they're actually at the emergency room and the doctor is trying to figure out why the patient has a fever, they will often do a blood count to see what the different types of blood cells are doing. And if neutrophils are high, that's going to be indicative of a bacterial infection. And so this is going to be objective 6 on your list, just to point you in the right direction. Now, monocytes and macrophages are very similar. They start out as the same type of cell. Monocytes, though, are going to be phagocytes that are simply going to be within the blood. That is where they are housed. And so once a macrophage moves out into the tissue, it's still a phagocyte, but it alters some of its surface proteins. So what it looks like to a scientist or doctors who are going to look at them, they alter some of their proteins on the surface, and then they are transformed into macrophages. Both monocytes and macrophages are phagocytes. They're just they have their different names based on their location. Monocytes are in the blood, macrophages are in tissues. Now there's also what are called histiocytes. Histiocytes are phagocytes. They are very similar to macrophages, but they're going to live in a certain tissue, and they're going to just stay there. And because of that, histiocytes / macrophages, they're going to have specific, or they're going to have other specific surface proteins to actually identify them as a particular type of macrophage. So, alveolar macrophages. Those are macrophages or histiocytes that live in the lungs. That's where they stay all the time. Kupfner cells live in the liver, or are found in the liver. Dendritic cells are phagocytes that are found in the skin, and that is where they live. And the macrophages are also called histiocytes if the macrophage stays in the spleen, the lymph node, the bone marrow, kidney, bone, and brain. So histiocyte, again, is the term used only if the macrophage stays in a particular tissue. So histio is tissue, cyte is cells, so they're tissue cells. But their purpose is to phagocytize. Alright, so what is the mechanism of phagocyte? Alright, they're going to recognize the foreign material, and the phagocyte is an eating cell, so they're going to phagocytize or physically engulf material, and in that way they can attack and dismantle foreign cells, and they can be isolated. Right? This can be an isolated event in terms of the dendritic cell randomly finding a bacteria cell that has gotten down underneath the skin and clear it, and it's all done. Or it can be part of an orchestrated event of inflammation, which we will see. So there are, depending on how you look at it, how you divvy up the actual steps of phagocytosis. Overall, there are 5 steps of phagocytosis. There's going to be chemotaxis, ingestion, the phagolysosome formation, destruction, and excretion. In your textbook, Table 12.1 shows phagocytosis as having 7 steps. And that's just because the figure breaks it up a little more. And so step 1, according to the figure, is just chemotaxis. And chemotaxis is movement towards or because of a chemical. And then the bacteria cells are right here. All right, and there is adhesion. So the bacteria cell and the phagocyte have to bind to each other. That binding process is going to send signals down inside of the host or inside the phagocyte. And it's going to cause this engulfment. And you don't actually see bacteria in this little invagination here, but imagine the bacteria are there. So there's engulfment, and then there is the creation of this phagosome. So the phagocyte takes bacteria in or other foreign material in by creating a phagosome. It's like a little pocket. And then step 5 here is what's called phagolysosome fusion or formation. And that's where your lysosomes here, which have different chemicals in it. There could be hydrogen peroxide in there. It could be oxygen radicals. It could be, interesting enough, small amounts of bleach. And so the lysosome and the phagosome are going to fuse in step 5 to create the phagolysosome. And by fusing, then these chemicals in the lysosome are released into the area that the bacteria is found. And then you have destruction, killing and dismantling of the bacteria cell. And then you have excretion. All right, so depending on how you look at it, it's best to be able to understand this 7-step process. That way you know you're not leaving anything out. But that is phagocytosis. And I think that is where we're going to stop for this section, and we'll start part 2 with the inflammatory response.